Thank you. All right, good afternoon. I hope you've all enjoyed your meal and you're all relaxed and ready for the afternoon session. My name is Bojan and I'm a lecturer here at UNSW Civil and Environmental, and I'm also project manager on the Nuisance and Harmful Algae Partnership with Melbourne Water. So this session is a continuation of the environmental impacts and control session from before the break. And our first speaker will be Olivia Daniels. So could I just ask Olivia to come over? And hopefully we can identify your slides.
That's the one. And the
This is the dangerous clicker.
Thank you. Hi everyone, I'm Olivia Daniels. Your program probably says that Laurel Fabro is giving this presentation. Laurel was going to present on my behalf, but I was able to make it to the conference, so I do apologise about any confusion there. Now, we did a research project on Limnothrix strain ACO243, and this one's a toxic cyanobacterium. And this particular part of the project, what we were looking at were light intensities, salinities, and temperature treatments. We wanted to see what it would grow at. And I should say, Laurel helps me with this, so did Sandrine Macchiella and Catherine Bernard. Now, this is roughly what I'll be talking about. I will talk a very little about the toxicology of it, or the toxicity. I believe the toxin has not yet been identified. So they're still trying to work on figuring out what it produces. Now, with the light intensity trials, these are the intensities that we tried. And you'll notice that we've actually got two treatments of complete dark. One of them with added glucose to the medium. Now, initially we weren't going to try and grow these in the dark, but one of our pilot trials went hopelessly wrong. So in our pilot trial, what we were doing was trying to purify or find an easier way to purify some of the cultures. And this was based on a method described by Vara et al. And essentially what you do is you cover 3/4 of a Petri dish after inoculating it, and in theory, these things being phototrophic are going to head towards the light, and you can isolate them because they move towards the light quicker than the bacteria and other contaminants will move. But unfortunately, after about 2-3 weeks of incubating these Petri dishes, we couldn't see any growth on the exposed area of that Petri dish. But lo and behold, when we took the cover off, there was a nice little colonies, and they were formed in the dark part of the Petri dish. So we thought, right, and anybody that's ever worked with Limnothrix, this particular strain is an absolute nightmare. Everything you want it to do, it doesn't. Everything you don't want it to do, it does. And this was one of them. So we thought, all right then, it obviously likes really dark conditions, so we're going to see if it'll grow in complete darkness. And we set up our experiment, and we had starting concentrations of 10,000 cells per mil, and had enough replicates and things like that, and all the replicates were inoculated from the same culture. And we put our cultures in a growth chamber and sampled every 48 hours and enumerated them. And these are the results that we got. Bear in mind that this is a log scale, but what we found that it grows fantastically at 160 micro-Einsteins. But we also notably got growth, although it wasn't an awful lot, we still got growth in the culture that was grown in complete darkness, providing that additional carbon source was available. And you'll see this bottom line here, that's the culture that was grown in complete darkness with no added carbon source. So it certainly didn't like that much. In fact, it died. But we did get growth with that additional carbon source. And for our temperature trials, these are the temperatures that we used, and they seem a little bit extreme, but Limnothrix has been found in ice caps, and it's also been found in hot springs. So we wanted to see whether or not this particular strain of Limnothrix could survive a vast condition of temperatures. Would have liked to have gotten the temperature a little bit lower than that, but unfortunately we're all limited by the equipment we can get a hold of. And the results, the methods, we used the same methods as we did with the light intensity trials. However, we did use a growth cabinet rather than the big growth chambers for these experiments. The results for the temperature trials, they showed absolutely fantastic growth, which you can see here. Again, this is a log scale, at 35 degrees Celsius. Really liked the hot water, and I believe that the Limnothrix out in the wild, I should say in the central Queensland region there, it's found usually in waters that are about 41 degrees Celsius. So they're usually shallow, but this stuff absolutely loves the hot water. Now, it did grow at 45 and 55, but we did not see as much growth. As I said, ideal growth at 35 degrees Celsius, which really shocked me. I wasn't expecting that at all. The problem with the temperature trials, as I said, Limnothrix does not behave itself. Now, D in the image there, that's a stock culture that was incubated at 27 degrees Celsius, but the others were at 7 degrees Celsius, and you can see here that the morphology, the colony morphology changes, and we have no idea what's causing that. Sometimes it forms these floating balls. Other times it grows, and that's fairly normal for the growth. But we also got these colonies here which adhered to the tissue culture flask, and it didn't matter how much you shook it, it was not coming loose. And we took a closer look of some of these colonies, and here's one I like to call hedgehog Limnothrix, because it looks very much like a little hedgehog there. And those holdfasts there, it formed these massive holdfasts. And I think for me the most interesting thing is when we had a closer look, what it forms is these almost like a skeleton. They are actually trichomes, and they're looped in a way to form an actual support structure. Now, this is quite possible because we also got photos of this stuff branching. It's not meant to branch. Again, this strain seems to do what it wants, when it wants. And we found a lot of these branches in the colonies. I know they're fairly hard to see here, but they don't grow very big at all. And Cormoric and Anagnostides, I hope I pronounced their names properly, did also report these small branches forming in Limnothrix. So I'm thinking, phew, it's not my imagination, I'm not going crazy. I suspect that these branches have formed to actually help it form those strange colonies that it does. And the advantages of that is it will survive nicely in a flooding event. Our salinity trials, they also shocked me. These are the salinities that we used in the trial. And interestingly enough, the best growth actually occurred with a little additional salt. But the methods, we used the same methods, and There's the results. They're all fairly similar, but again, this is a log scale. But what you'll see is that 1 gram per litre of sodium chloride really helped this stuff grow. It loved it. In fact, the growth was similar or it was better with that additional salt than it was in just plain fresh water. Didn't like the really heavy salt, but having said that, it still grew, just not very well. Now, the interesting parts about in the heavy salinity concentrations, what we actually saw was the cells becoming slightly rounder, and what they were doing was breaking off the trichome and forming these short little clusters of almost spherical cells. They were quite bulbous at each end, and have no idea why they were doing that, but they were doing that. Now, the conclusions for this particular research is it grows very well it grows in the dark, providing there's an alternate carbon source available, which is quite relevant because we're starting to find Limnothrix in drinking water pipes and just water pipes in general, which is a bit of a concern considering it's quite toxic. It grows in saline conditions and at relatively high temperatures. Now, I also did some experiments with fungi and this particular strain, and it grows brilliantly with fungi as well. I was expecting there to be some allelopathy or something going on, but no, it actually grew better with the fungi than it did without the fungi. And everything that we looked at, kind of suggests that it would grow really, really nicely in the soil around the edge of the water. And, because it can tolerate that higher salinity that you would find at the edge there, and also the warmer temperatures, and, of course, the fact that it grows in the dark and forms those strange colonies, which would help it, if the water levels were going up and down. Now, the problem is, as I said, it is very toxic, and, I know there have been cattle deaths as a result of this particular strain. One of the things is if it is in the soil around that water edge, it could be easily transferred, from the hoofs of animals from one watering hole potentially to another. But it can also impact sensitive species. Now, I did a lot of toxicity trials with cane toad tadpoles, and bear with me, I don't know if I can get this to play, but I managed to capture one of the tadpoles actually having a convulsion. Now, there were no signs of it being sick beforehand, and unfortunately, a lot of the tadpoles were actually doing this. They were having convulsions and then just dying soon after exposure. Let's see. No, I don't think it's going to play. No, which is... but the image is a bit blurred because, as I said, it was just by chance we had been filming. But it's on USB, so if you want to see it afterwards. It's only 12-second footage. But histology showed that the toxin did a massive amount of damage, and the damage was to every organ that we examined, including the notochord, the brain, the whole lot. You couldn't recognise the organs after the toxin had finished with it. Now, here on the left-hand side, what we actually looked at was this toxin while aerating the flasks that the tadpoles were in. And we also looked at the toxin without aerating the flask to see if that low dissolved oxygen would make a difference, and it certainly did. On the left-hand side, these three images, they're at different different live cell concentrations, A being the control, And E is at 100,000 cells per mil, and that's the cell concentration that we find the blooms in quite often, and that happens to be the concentration that did all the damage. I did take it to a higher concentration, but it seemed to be that 100,000 cells per mil that did all the damage. And it was significantly worse when the flasks were not aerated. I mean, that, believe it or not, is part of the stomach, and you can see that it's just completely seized up. There's, necrotic cells everywhere, and it was just a mess. The brain was almost completely disintegrated in the animals. It was really, really awful. That toxin made a mess. And that actually sums that up, and thank you very much for listening, and if you have any questions, feel free to ask away.
Olivia, that was bang on time, so we have plenty of time for questions. Would like to start. Do you have any idea what the toxin might be?
No.
It's not telling us.
Well, I have an idea, thanks to Laurel, but because it's not, I didn't do any of the toxicity studies. I'm not in a position where I can say what the toxin is. So yeah, there's probably, just bail Laurel up. So sorry, I'm claiming I don't know. Yeah, it is very nasty though.
I have a question.
Yes.
You're claiming that it's growing in pipes, and I was just wondering if you could comment whether they were chlorinated pipes or not. So what chlorination levels?
To be honest, I don't know. Again, that was one of the fellow researchers, Adam Rose. He's been tracking it, and having said that, I should say that he's identified Limnothrix in the pipes, not necessarily this strain, but the way this particular strain grows, it certainly wouldn't surprise me if it can actually tolerate chlorination. But again, to be honest, I don't know if and again, Laurel's probably the best person to ask about this. Do you know if they're doing any chlorine studies on it?
Salmon that is growing in pipes between 1 and 4 ppm of environment.
Ah, Noel found them in pipes. Wow. So, yeah, so there you go. It can handle the standard chlorine treatment. Yeah.
All right. Thank you very much. Please join me in thanking Olivia.
Thank you.